Okay, so let's start with this last session of the workshop. So this post session from students. We have received 5 different posters, so let's start with the presentation of the first one. The first one, it's the work, it's entitled Effect of Biochar and Sheep Manure on Maize Plants Grown in Contaminated Mine Soil. The corresponding author is Taoufik El Rasafi from the University of Sultan Moulay Slimane in Morocco. So, Taoufik, the floor is yours. It's not connected. Oh, yeah.
Hello everyone. I hope you are seeing my screen and hearing me well.
Yeah, that's all correct.
Um, First of all, I want to thank you very much for giving me the opportunity to be with you today and present the resume of my work. I want also to say hello to Prof. Pereira. It's a nice pleasure to see you again. So my work today, entitled Effect of Biochar and Cheap Manner of Maize Plant Grown in Contaminated Soil. Uh, First of all, I want to present myself. My name is Taoufik El Rasafi, PhD in environmental engineering and plant biology. I just finished a postdoc experience in one of the latest Moroccan universities, which is University of Mohammed VI Polytechnic in Ben Gurir. I had different experience of workshop and trainings, starting from Germany in the laboratory of Professor Jörg Rombok, which I thank him for everything and for the nice period in his lab. Also, 2 experiences in the laboratory of Professor Ruth Pereira and her supervisor, and that of Catarina Marquez Ribeiro. I want really to express my thanks for their help, for support, for everything they have done for me during this experience. Also, I had 2 experiences in China. The first concerned high efficient plant factory technologies, and the second regarding rainwater harvesting. Uh, Generally, my work focused on toxicity of metals on plants and the wind tests starting from germination and early seedling growth to tests in pots and in fields. So, as you know, metals trace elements are arrived to soil from different activities such as industrial activities, urban chairs, agriculture, and mining activities. Those elements can be transferred to food chain, to ecosystem, water, resource, and affecting by the end plants, animals, fauna of the soil, and humans by direct or indirect injection. So the problem here is how to remediate this problem, how to decontaminate those soils. As you know, in Morocco, now there are more than 240 mine sites which are active and more than 200 sites which are abandoned. So this led a big amount of lands and wastes which are very toxic and contain a lot of amount of trace elements. The site which we studied, which is the site of Ait
Sorry,
Ammar.
Taufik, I think that we are only seeing the cover of the presentation. If you are passing the slides, we are not seeing.
Yes, I passed.
So we are not seeing. It's freeze the image. Try to stop and start presenting again.
Okay.
Right now, I think we are seeing the.
The
Now we
first
are
is
seeing. Yeah. Now, no. Now it's working.
Now it's working. And
Yeah.
you see it in all the on the screen, or only?
It's not completely the screen. It's full screen. Click on here. Yeah, I think.
Now it's moving.
No, it's not moving.
And now, okay, you
Now,
see?
yeah. Now, yeah, it's moving.
Okay, so okay. So I said that I spent a lot of experience of workshop and training in Portugal, Germany, and China. Also, this is regarding the work that we have did about metal toxicity, starting from germination and early seedling growth to pot experiments, and we are now preparing some papers regarding the field tests about toxicity, also some works about microorganisms and their ability to solubilize phosphate rocks. Here is the general main sources of metal toxicity, agriculture, mining activity, and so on. And the problem is how to depollute and decontaminate the soil. Here is a picture showing that a lot of mine sites are active and abandoned in Moroccan soil, which led a lot of waste and lands contaminated with a lot of amount of trace elements. So the work that we are presenting here is a work done in the iron mine site, which is abandoned since 1962. This work is had the object of different tests, starting from characterization and physico-chemical composition of the soil, bacteria, effect on human health, animals, incitridis, arcarians, and also accumulation of plants in native sites. Those works are under a project to NATO sites for peace, which is which were under the coordination of Professor Root and other professors. So after that, we thought that we need to find a solution to depollute the soil. So that's why we choose to use organic amendments as a solution, and the main object here is evaluate the effect of biochar and animal manure as soil amendments on morphological attributes of maize grown in this mine soil. So here we took biochar prepared in the traditional way and animal manner, then grounded and saved. We took we used two concentrations, 2.5 and 5% of biochar and 10% and 20% of animal manure, mixed thoroughly with the soil and left for incubation for 10 days. After that, the seeds of maize were added to soil and left for growth for 30 days in normal and ambient environmental conditions. In the end of the experience, we measured the morphological parameters of maize seeds, such as root elongation, leaf growth, leaf number of leaves, and so on, leaf area, dry biomass, and also we characterize the soil by measuring the pH and the electrical conductivity. So the results shows some good results. The soil pH in presence of the amendments increased comparing to the soil as you see in the beginning of the test. Also, electrochemical conductivity decreased in the contaminated soil compared to that found in soil before the start of the test. By adding animal manure, this electrical conductivity was increased higher than that in the end or before starting the test. However, electrolytic conductivity in presence of biochar was decreased. Regarding plants, we not observed any dead plants in the treatment. We had also the plants showed us some few signs of phytotoxicity, which is induced by the contaminated soil. As you see in the picture, in the contaminated soil, the leaves were small, brown sheet, and mid-rape, showing irregular margins, and their colors start to be low. Will the leaves in the soil amended with animal manure, especially animal manure, or were larger, green, and longer. Those signs and suggest that biochar and animal manure reduced physical symptoms of metals toxicity. Regarding the other parameters, we see that no significant differences among root longitude, but some significance were observed in shoot elongation, leaf longitude, and leaf number. Also, no significance was observed in root dry biomass, and the use of different treatment has led to a significant increase in dry matter of the stem of maize, with the roots who had were the same in all the treatment. Regarding leaf area, the lowest leaf area is found in plant grown in contaminated soil, and the highest were observed in biochar treatment and animal manure treatment. We also ran some persons correlations and principal compound analysis. We found three principal compounds. The first one is around 47% of the total variance, and which is strongly associated with diameter, leaf number, and dry weight of shoots and leaf longitude. In the end, the use of organic amendments in soil contaminated with potential toxic elements is very important and effective, and gives positive effects on corn growth. Also, the morphological plant traits such as plant height, stem diameter, and other parameters have been improved after addition of both biochar and animal manure. Regarding concentrations, we observed that 10% animal manure and 5% biochar showed positive effects on the majority of the parameter measured. And the organic amendments can be used in enhancing the phytoremediation of contaminated sites. This work is evaluated and published in biomass conversion and biorefinery in 2021, and we are preparing another about translocation, uptake, and root architecture, which I thank Professor Abdullah Okaroum, where we did all those measurements in lab of Professor Abdullah Okaroum. And thank you very much for your attention.
Thank you very much, Taoufik. So let's open now the discussion questions from the audience.
Well, I can start. First of all, I would like to congratulate Taoufik, and it's nice. It was a good surprise to see your name in the list of posters. I don't know nothing about you for some time, but it's nice to go that it's nice to know that you are going on your research activities and producing good papers and good work. So congratulations for that. And I just would like to know if you are planning, because it could be interesting to assess the bioavailability of metals in the soils. If the applications of manure and of biochar are changing the mobility of metals in soils. And just another question, do you think it's feasible to, because I know the place, as you know, and do you think it's feasible to start planting, to convert this area for an agriculture area, because it could be good for people that lives nearby?
Thank you very much, Professor Roth. It's really nice also to see you after all this time, and I take chance to thank you very much for all the effort that you had did for me during the experiences that I spent in your lab. Regarding your questions, we are planning. We already did the testing, the accumulation and transport and translocation of elements from soil to plant. We are preparing another paper regarding that. Regarding the rehabilitation of the mine soil, I think it's a little bit difficult. As you know, there is mountains and some soils that not good for culture, but it's better to select some area with the plan area, than start to take some trials to see if there is a translocation and to plants. Then we can use it after 1 or 2 years, or maybe depends of the results. Regarding the mountain areas, we can use some trees, which could be better to explore those soils.
Thank you very much. Congratulations once again, and go on.
You're welcome. Thank you very much. You're welcome.
So the experiments, let me do is just curiosity, because sorry, I'm not from this this research field. I'm an engineer, so for me it's really new, that part. But you have studied in laboratory. You haven't done any experiment on a real contaminated mine soil. So it's just with the plants that we have seen in the photographs you presented in the presentation.
No, until now we just made tests in the laboratory. Then, as Professor Root said, maybe after we plan to use or to test some field tests in the contaminated soil. Okay. The field, it's a little bit difficult to control all the conditions. Climate, other animals, maybe the people living around there. So it's a little bit environmental conditions also. It's a little bit complex to control all those conditions. That's why we are planning to do it, inshallah, in the future.
Okay, so congratulations for the work, and thank you.
Albert,
yeah,
if I may.
Yeah, sure.
Yeah. Thank you. Thank you, Taoufik, for your nice presentation. Are you planning also to use sensors in your environmental lab to measure this the parameters as you mentioned, like leaf area index, maybe the stress, the color variation in the leaves, like thermal sensors, hyperspectral sensors, or even RGB sensors can be used, simple RGB sensor?
Until now, Professor, we or the tests we used only the morphological tests by the traditional way, either by using e-tools to measure the leaf area or visual change in the color. But maybe in the future, in collaboration with you, we can use
Well,
those.
certainly it would be of interest and related to the topic of TERRATECH
Yes, it will.
to
It will be a
to
pleasure.
use the sensors, you know, and then then you have digital, you know, data in which you can measure. And the nice thing would be with the data, then somebody else can repeat the, you know, the experiment.
Yeah, it
And
will be a pleasure for us
yeah,
to do this together. It will be
certainly. No,
yeah.
would be great. Thank you.
You're welcome. You're welcome.
Any other question to Taoufik? If not, congratulations again for the work and the presentation, and thank you for joining.
Thank you very much, and thank you for giving me the opportunity to be with you today. Thank you.
You're welcome.